So today we're going to dive into a really groundbreaking piece of technology that's helping us fight one of our oldest enemies, the flu. We're talking about a lung on a chip and how it's literally helping us crack the code of what makes influenza so severe.
Let's just start with a number, a really big one, over 300,000.
That's how many people die every single year from severe influenza. It's this relentless global threat that, honestly, continues to challenge even our most advanced modern medicine.
And that leads to this incredibly frustrating question that scientists have been wrestling with for ages. We develop so many promising treatments in the lab. They look great, but when they finally get to human clinical trials, they just fail. So why? Why is there this massive gap between our lab models and what happens in a real person?
Well, to answer that, we first have to really understand the core of the problem, which you might call flu's deadly puzzle.
So here's the heart of the matter. For decades, we've had to rely on animal models, mice, ferrets, and simple cell cultures in a petri dish. But here's the thing. They're just not great substitutes for a human being. They have different receptors for the virus, their blood vessels are simpler, and their immune systems just aren't as complex. You know, testing a drug on them is kind of like trying a key in the wrong lock. It might almost fit, but it's not going to open the door.
And the result of this mismatch is, well, it's staggering. Get this. Only about 12% of therapies that look promising enough to enter clinical trials ever actually make it to the market. That's not just a scientific curiosity, it's a multi-billion-dollar bottleneck in medicine. Think of all the wasted time and resources that could have gone into treatments that actually work.
So, if the old models are broken, what do? You build a better one. And that brings us to the real hero of our story today, the tool that's making all this possible. It's, quite literally, a living human lung on a chip. It's officially called the Immune Competent Lung on a Chip, or IC-LOC for short. And yeah, it is exactly what it sounds like. A tiny micro device about the size of a computer chip that contains living human lung cells and immune cells, all arranged in this beautiful 3D structure that perfectly mimics a small human airway.
Building one of these it's a masterclass in bioengineering. It's this incredibly meticulous process that takes over 9 days. It all starts on day 0, when they lay the foundation, seeding a vascular layer with blood vessel cells and structural cells. Then by day 4, they create this crucial air-liquid interface. And what that does is it basically tells the airway cells, okay, time to grow up and become a real lung. But they're not done yet. On day 7, they add specialized immune cells, macrophages, to the top layer. And the final amazing piece of the puzzle comes on day 9, when they perfuse the tiny blood vessels with circulating immune cells from actual human blood. That's what brings the whole system to life.
So the result is this incredible little device that contains the whole shebang. You've got the lung lining, the structural cells, the blood vessels, and a complete set of both the immune cells that live in the tissue and the ones that circulate in your blood. It's not just a model anymore, it's a tiny, living, breathing system.
Okay, so you've built this amazing mini human lung. Now what? Well, now comes the real test. Can it recreate the absolute deadliest part of a severe flu infection? We're talking about the infamous cytokine storm, where the body's own immune system just goes haywire and starts attacking itself.
And the answer was a spectacular, resounding yes. Look at this. When the researchers measured a key inflammatory signal, a simple chip with just lung cells barely reacted. But the full immune chip? Whoa. It lit up like a Christmas tree, producing inflammatory signals at levels that were nearly identical to those found in the lungs of actual, critically ill flu patients. This was the aha moment. This was the proof that they had built a model that didn't just imitate the disease, it experienced it.
I mean, you don't have to take my word for it. The researchers themselves put it perfectly in their paper. The IC-LOC system recapitulates the hyper-inflammatory environment observed in patients with severe influenza. In other words, it's the real deal.
So once you have a model that's this accurate, you can finally use it like a detective to find clues and start identifying the real key players driving the disease.
And what the chip revealed was just incredible. First, they saw that the immune cells were actually protecting the lung structure from being totally destroyed by the virus. Second, and this is a big one, for the very first time in any lab model, they actually watched circulating immune cells crawl through the vessel walls to get to the infected tissue. And most importantly, the model let them pinpoint the exact molecular signals that were driving this dangerous overreaction.
They zeroed in on one key pathway in particular, something called the CXCL12-CXCR4 axis. The easiest way to think of it is like a chemical emergency flare, a powerful get-over-here-now signal for immune cells. And what was so fascinating was that this flare wasn't just being shot up by immune cells, but by the lung's own structural cells, which showed that this was a much deeper, more complicated response than anyone realized.
Okay, so identifying a suspect is one thing, but finding a weapon to actually stop it, that's another challenge entirely. And this is where the lung on a chip just absolutely shines, leading to a really surprising revelation.
The researchers then tested several potential drugs on these infected chips. Tamiflu, the standard treatment we use today, actually made the inflammation a bit worse. Whoops. Other drugs had mixed results, some even let the virus run wild. But then, they tested a drug specifically designed to block that CXCR4 pathway we just talked about, and the result was amazing.
The CXCR4 inhibitor was totally unique. It was the only drug that did both things you needed it to do. It calmed that dangerous cytokine storm, and it helped reduce the amount of virus in the lung. It wasn't just killing the virus, it was changing the whole environment to be less self-destructive and more antiviral. A total game changer.
So what does all of this really mean for the future? Well, this goes way beyond just the flu. The real impact here is that the future of discovering new drugs is finally becoming truly human relevant.
Honestly, this technology changes everything. We now have a faster, more accurate way to study not just flu, but all sorts of complex lung diseases. It means we can finally drastically reduce our reliance on those animal models that so often lead us down the wrong path. It opens up this whole new world for critical research on how pathogens interact with our bodies, and it gives us a platform to discover entirely new drug targets for so many diseases.
And that brings us to a final, really powerful question. The immune cells that were used in this study all came from a single person. So just think about it for a second. If we can now build a mini lung using your cells and test which drug works best for your specific immune system, are we finally on the verge of truly personalized medicine? It's a future that, thanks to tech like this, might be a lot closer than any of us think.